Biosensors. First Biosensors அப்படிங்கிறது என்ன அப்படிங்கிறத பார்ப்போம். What is mean by the biosensors? Biosensors அப்படிங்கிறது it is used in the research nowadays. It has many advantages in the various fields like nanobiotechnology and biometrics, synthetic biology, which has led to the combination of many new fields like silicon technology and for the development of the bioreceptors which are used nowadays. Next, there is the interface architecture which provides an environment for the proper functioning of the biosensing elements, which gives rise to physical phenomenon that takes place. The physical phenomenon or chemical response is exhibited by the analyte molecules. What is mean by the analyte molecule? The analyte molecule is the molecule which binds to the substrate, that is called as the analyte molecule. This is the membrane S-type biosensors. This is one of the biosensors which is present in the lipid bilayer membrane, which is usually present in some of the bacteria. The biosensing element, the common materials which are used as the biosensing elements include the living cells, tissues, enzymes, membrane proteins, receptor channels, pore membrane proteins, and active peptides. Antibodies are usually used, and the samples which are used for the as the bioreceptors include the blood, urine, saliva, tears, and various secretions are also used. We can also check the biosensing ability of the food samples also, food samples as well as the environmental samples. Biological molecules may aggregate on the surface of the electrodes or the sensors which bind to the particular solid substrate. When it is the ion sensitive, when the particular molecule is ion sensitive, it binds and reacts with the particular biosensor. And the intermediate layer which is present in the biosensor, it consists of the various polymers like PEG, chitosan particles, agarose, hydrogel are usually present. It has also got the self-assembled monolayers which are present on the surface of the biosensors, and which has the monomolecular array of self-assembled proteins are usually present. The lipid membrane is used as the biosensing element. Sometimes when the molecule does not properly bind to the analyte, it is called as the incomplete mode of biosensing ability. This arrangement facilitates the many characteristic features of biosensing ability. There are some certain examples which has been reported by various researchers. Pitcher et al., he reported that the phenomena which is exhibited by the amphorometric sensors coated by 4 layers. The metal used is usually the zinc oxide, along with the nanoparticles which are used, or gold nanoparticles or silver nanoparticles are usually used in this type of methods. Some of the membrane active peptides are also reported from the various microorganisms. Gramicidin from Bacillus brevis, alamethacin from Trichoderma viridae, valinomycin from Streptomyces species, PGL, that is the peptidyl glycine leucine carboxamide, which is synthesized by protein chemistry. and alpha hemolysin, which is reported from Staph aureus. Voltage dependent anion channels, they are located on the mitochondrial membrane and they also act as the biosensors. Lipid bilayer based biosensors are also another type of biosensors which works on the principle of Coulter counter principle, which is based on the size and number of the analyte molecules which bind to the efficient targets. Anatomy of the fluorescent biosensors. Then what is the anatomy is the it consists of the biomarker and it reacts to the biomarker and the, the particular diseases are diagnosed with the help of the biomarker. This is the protein biosensor usually consists of the analyte which binds to the fluorescent probes. These are the various environmental factors. There are some environmental factors which influence the biosensing ability and binding efficiency of the biosensor. It is usually the solvent accessibility, changes in the ionization of the dye, fluorescent resonance energy, and protein structural prediction and protein structural changes which is exhibited by the characteristic biomolecule. That the biomolecule may be any protein or any novel compounds which are isolated from the microorganisms or any compounds which are isolated from dye degrading microbes or any other compounds which we isolate out of our research work. Thank you for your patience. Thank you.